Welcome to the new lectures of Bio 110. This set of lectures, we will be looking at the bioenergetics. So this first lecture will be used for Monday's class on September 29. So what I have in my first slide here are the set of objectives that I would like you to accomplish in chapter 13. I am not going to read every single objective for you right now, but I will have them here for you to review. As we did in the previous exams, these objectives will be the basis for testing in the class. Alright, let's begin to explore how the cell produces energy that it's going to use for its cellular processes. The first thing that we have to remember, it's the enzymes that the cell produces for its metabolic pathways. We're going to be looking at glycolysis. We're going to be looking at the citric acid cycle, or also known as the Krebs cycle. And we're going to use that information for Lab 3 that will be coming up in a couple of weeks. We're also going to look at aerobic respiration, which is how the cell uses the electron transport chain to derive energy in ATP when oxygen is present. And we're going to look at the end at anaerobic respiration, or basically how the cell derives energy when oxygen is absent. One of the methods that we're going to be looking at will be also fermentation. I would like you to also review some materials from chapter 3, issues about metabolism, anabolic and catabolic pathways, the concept of free energy, activation energy, what a catalyst is, the energy carriers such as ATP, and the electron carriers such as NADH and FADH2. Also, please review coupled enzymatic reactions and what it means to have an oxidation and reduction reaction. So remember, in Bio 2 you learned that food molecules are going to be broken down through catalytic pathways, and those catalytic pathways are going to generate useful forms of energy, usually in the form of ATP and electron carriers. You're going to generate building blocks that could be used to build other molecules. This will be, for example, amino acids, simple sugars, nucleotides. And you're going to lose some of that energy that is derived from the catabolic pathways as heat. Now later, in the anabolic pathways, you can take some of that energy that you generated by breaking down the food molecules, ATP or the electron carriers, and use it together with the monomers of those building blocks that you generated to generate macromolecules that the cell will need. So in that way, you can go and recycle the energy and molecules that you obtain from food into making new molecules that the cell may need. Now, these pathways are going to happen through basic metabolic reactions. So things that I would like you to remember are going to be the enzymes and substrates and metabolic intermediates. So in this particular image, what we have is a substrate, molecule A, that will be taken by enzyme 1, and enzyme 1 will catalyze the reaction to make molecule B. Molecule B will then be used by enzyme 2 in a catalytic reaction that will generate molecule C. Molecule C will then be used by another enzyme, enzyme 3, in a catalytic reaction to generate molecule D. And the same situation will happen with enzyme 4 and molecule E, and enzyme 5 to the final product, molecule F. So in this pathway that we have here, molecule A is the initial substrate of the reaction, molecule F is the final substrate of the reaction, and molecule B, C, D, and E are the metabolic intermediates. So just as a review, I would like you to remember and give me an information. How many genes are involved in this particular metabolic pathway? And we can have this discussion on Monday. One issue that I would also like you to remember is the concept of activation energy. What we have here in figure A, it's a graphical demonstration of a reaction. The reaction is taking molecule Y to molecule X. Now, this reaction may happen spontaneously, but in order for molecule Y to become molecule X, you will have to overcome an activation energy, shown over here by this peak. So that energy goes from B to A, and the energy of the system has to overcome that activation energy to allow molecule Y to become molecule X. In a catalyzed reaction, in a reaction catalyzed by an enzyme, please notice that the same reaction, molecule Y being catalyzed to molecule X, now the activation energy has been dramatically reduced. The enzyme is able to lower the activation energy, allowing the reaction to proceed forward. So the enzyme is the catalyst that is going to be, the catalyst that is going to help mediate the reaction Y to X. And the enzyme's product will be, the enzyme's function, will be to overcome the catalytic reaction. Now, depending on the kind of reaction that you are studying, some of those reactions can have they can happen spontaneously because they're energetically favorable, and other ones may not happen spontaneously because they're energetically unfavorable. In an energetically favorable reaction, in here for example, again, Y becoming X, this is a catabolically energetically favorable reaction because the free energy of molecule Y is greater than the free energy of molecule X. Therefore, the change in free energy is going to be less than zero, giving us a negative value. Because the energy then it's released from the system that is going to increase the disorder around the environment, which helps drive the reaction forward. So in a spontaneous reaction, you're going to have a negative delta G and an increase in disorder. Now, if we were to think about the opposite reaction, molecule X becoming molecule Y, because the free energy of molecule X it's lower than the free energy of molecule Y, the change in free energy it's going to be positive, and therefore that reaction is energetically unfavorable, and it will increase the order of the surrounding environment. So that reaction, in order to happen, will require the input of energy in order to allow the reaction to move forward. So that reaction in a biological system can only occur if it is coupled to a second reaction that is energetically favorable. Now, let's think about how the reactions are going to be thought up when we have the product of glucose coming into the cell to produce and give energy to the cell. So the main players involved in this scenario will be the glucose molecule. You're going to have ADP, or you're going to have the energy carrier ATP. You're going to have phosphorylated intermediates during the reactions. You're going to have activated electron carriers, in particular NADH, and later we're going to see FADH too, and that will happen in the Krebs cycle. And therefore, of course, you have enzymes that are going to lower the free energy, lower the activation energy, of the reactions to allow them to happen. So if we were to write this reaction as an equation, we'll have a glucose molecule, 6 oxygen molecules, O2 molecules, and by a set of catalytic reactions, at the end, we're going to have the complete oxidation of glucose into 6 carbon dioxide molecules, 6 water molecules, and energy. Now, the delta G, the change in free energy for glucose becoming carbon dioxide and water, it's going to be negative 686 kilocalories per mole, indicating that the reaction can happen spontaneously and liberate energy. So how is glucose stored in animal cells? In animal cells, glucose is stored in the form of glycogen. Glycogen is a polymer of glucose, and the polymer of glucose involves long chains of glucose molecules which are bound to one another by an alpha 1 to 4 glycosidic bond. But those long chains can have branches, and those branches will be generated by having other chains which are joined together by an alpha 1,6 glycosidic bond. So glycogen, being a polymer of glucose, it is a great way for glucose molecules to be stored in the animal cell. Similarly, in plant cells, what you have is starch. Starch is another polymer of glucose that is used to store large amount of glucose. For the purpose of our class, we're going to look at how glycogen stores glucose molecules as long carbohydrate polymers, and those glucose molecules then can be used one by one to generate energy for the cell. Here in this other image, what I want to show you inside this liver cell are glycogen granules, and those glycogen granules are ways in which the glucose is stored for later use in the liver. Now, the glucose molecule on its own, it is a ring. Here in this particular image, I am showing you the nomenclature of each of the carbon molecules in the glucose molecules. Glucose has 6 carbons. Carbon 1, carbon 2, 3, 4, 5, and this one, which is branching outside of the ring, carbon 6. Depending on the way that you depict the molecule, you can see it as a stoichiometrically moved ring. It's not a flat ring like a benzene ring, but it actually has geometry to it. And here at the bottom you have a stick figure of it, as well as a space fill model of the glucose molecule. But what I would like to remember, when you're looking at a glycosidic bond in glycogen, if you go back one step, the glycogen molecule, it stored the alpha 1 to 4 glycosidic bond. That means that carbon 1 of the neighboring glucose molecule is connected to carbon 4 of the other glucose molecule. And in the branching point, you have now carbon 1 of a glucose molecule connected to carbon 6. And this is why I brought up to you the nomenclature of the carbon, so you understand the position and why it is an alpha 1 to 6 or alpha 1 to 4 glycosidic bond. The alpha denomination means that the molecules are on the same side of the bond. Now, as a review, I also want to show you the concept of ATP hydrolysis. ATP, it is a nucleotide. The nucleotide contains a base, adenine, and a 5-carbon sugar, ribose. Now, that will be the nucleotide that is put together. The nucleoside will be adenosine, and adenosine has 3 phosphate groups attached to carbon 5 of the ribose. Now, between each of the phosphates, you have a phosphoanhydrate bond that is a high energy bond. So if when broken, when ATP, for example, is hydrolyzed to ADP, an inorganic phosphate, abbreviated PI for inorganic, you can release the energy of that phosphoanhydrate bond to use in a cellular reaction. Later, the cell will be able to take ADP, or adenosine 5-diphosphate, and ADP plus an inorganic phosphate. If you spend energy, you can then add back the phosphate. So one reaction, the ATP for ADP, an inorganic phosphate, has a negative delta G, so the reaction can happen spontaneously and generate energy. But the reaction of ADP plus inorganic phosphate to ATP has a positive delta G and therefore requires that energy be given to the system to occur. Now, in many of the glycosidic reactions, what you're going to encounter is the donation of a phosphate to a molecule, and that is considered substrate level phosphorylation. And substrate level phosphorylation can generate phosphorylated intermediates that carry energy, and that energy can later be used to harvest ATP or to catalyze a reaction. So for example, in an activation step, substrate B, which contains a hydroxyl group, the enzyme will catalyze the hydrolysis of ATP into ADP and substrate B with a phosphorylated intermediate. So now B phosphate can later be used by another enzyme, and that enzyme can take, in this case, for example, substrate A, which has a free hydrogen group, and in a condensation step, use the energy of the removal of that phosphate to generate the product AB. In that way, through substrate level phosphorylation, you can use the energy harnessed or harbored in that phosphate bond to catalyze the reaction substrate B phosphate plus A to product AB. Also, not shown here, that reaction could be used to generate ATP by using ADP and the phosphorylated substrate to generate ATP, and that is going to be the case in glycolysis. So now, to illustrate the principle again, we have here a substrate that contains a hydroxyl group, and that molecule, the enzyme will then be able to break the phosphoanhydrate bond between phosphate 3 and phosphate 2 of ATP. That phosphate transfer will have a negative free energy delta G, change in free energy, and therefore generate an ADP molecule together with the phosphorylated molecule. So here will be the molecule with the phosphate attached to it. That energy with the phosphoester bond will be able to be used to generate ATP again, or to give the energy required for a different reaction. Now, another concept that I would like you to remember, it's the issue of an oxidation and reduction. When glucose is broken down, the molecule is being oxidized because the molecule is going to donate electrons, and the electrons are donated together with hydrogens. On the other hand, the oxygen molecule is going to be reduced because that molecule is going to gain the electrons, of course, and also its hydrogens to generate water. So, as a review, remember that oxidation is involve the loss of electrons. Reduction is going to involve the gain of electrons. Now, when we think about an oxidizing agent, it's going to be the molecule that is going to be reduced. It's going to be the electron acceptor. In this case, it's going to be oxygen. The reducing agent is going to be the electron donor, or the molecule that is going to be oxidized. And in this example, is going to be glucose. Now, those electrons are usually carried by activated carrier molecules. Illustrated in this image from chapter 3, what we have here as a truck, it's the carrier molecule that is going to be moving the cargo of energy back and forth between two different reactions. So for example, here you have a food molecule, and that food molecule is carrying energy. That energy is being carried between the bonds of the food molecule, in particular the carbon bonds. In an energetically favorable reaction, that food molecule will be oxidized, meaning that it's going to be donating the electrons or donating the energy in this case. So this empty activator carrier molecule, shown here by the empty truck, it's now going to be able to carry that energy to another molecule that is going to be involved in an energetically unfavorable reaction. So you have this molecule here available in the cell in an anabolic reaction, and that molecule will then be able to use the energy brought in by the activated energy carrier to generate a new molecule that carries energy. So in the left, we have the catabolic reactions, and the catabolic reactions are going to be the ones in which the energy will be harvested. And on the right, we have the anabolic reactions, in which the energy harvested from the molecules on the food are going to be used to make new molecules that require energy for building. Now, one of these electron energy carriers is going to be FAD, flavin adenine dinucleotide. So FAD is an electron carrier that is going to be reduced to FADH2. So FAD, it is a molecule containing nucleotide, adenine diphosphate, and you have a component which is a riboflavin, which is vitamin B2. The riboflavin has a flavin molecule, and in the flavin molecule it's composed of 3 aromatic rings. Now, highlighted in yellow over here, you have the atoms that are going to be involved in the reduction reaction. So FAD will be able to take 2 electrons and 2 protons, and what you can observe in the yellow boxes, it's the breaking of the double bond between nitrogen and carbon to admit 1 hydrogen, the breaking of the lower double bond between nitrogen and carbon, accepting the other hydrogen, and the generation of a new double bond between the 2 carbon molecules. So this happens sequentially. So only 1 electron will be accepted at a time to have an intermediate molecule that has an FADH. And later the other electron and hydrogen will be accepted, generating the final product of FADH2. Another activated electron carrier is NAD. And NAD is going to be reduced to NADH. NAD is the nicotinamine adenine dinucleotide. Like FADH, it has kind of the same structure. You have nucleotide, adenosine dinucleotide with 2 phosphate, and that is put together with another nucleotide which has a nicotinamine ring. Nicotinamine, here shown in the left, is the amide form of nicotinic acid, which is the vitamin B3, or also known as niacin. So now, in a reaction that is going to involve a hydroxyl group in a molecule, that hydroxyl group is going to be able to donate electrons to the NAD plus molecule, or here shown as NADPH, which has a phosphate group in the ribose. So it's the same kind of structure, except that there's a ribose, there's a phosphate group in the carbon 2 of the ribose. That's the only difference between NAD and NADP. When the hydroxyl group, back over here, donates its electrons, NAD will be able to accept it, and that will generate a carbonyl group in the donor molecule. Now, NAD is going to be accepting the electrons, so it will be NADH or NADPH if it's in a plant cell. And later, NADH will be able to donate that electron in a subsequent redox reaction. So in the example here, what we have is an alkene, which is a double bond between 2 carbons, and that molecule will be able to receive the electrons and be reduced, and therefore now you have a generation of a single bond between the 2 carbons, which is a saturated molecule now. So you have each carbon bound by a hydrogen. So in the lower portion of this molecule, what I want to point out is the fact that in the nicotinamide ring, you have a shifting of the double bonds within the ring to accept the hydrogen in the carbon that is adjacent to the nicotinamide moiety, shown over here. Now, let's take a look at glucose. And let's take a look at how the energy of glucose can be harvested. Outside of the cell, glucose in an oxygen can be burned to generate carbon dioxide and water. That reaction doesn't happen spontaneously because it has a very large activation energy. But if you were to take glucose and start to apply heat to it, you can overcome the activation reaction that is going to eventually lead to a great release of energy and the breaking of the bonds between the glucose molecules, and therefore generate CO2 and water. All that energy, when you burn sugar, for example, it's released as heat, and none of them is stored. It's given to the environment. Just try making caramel out of sugar. You will see that it will go from a solid state to a liquid, but eventually all of it will become burned carbon if sufficient energy is applied to the system. That will be a great waste for a cell, if this will be the way that the cell harvests the energy. So in order to harvest the energy of all independent carbon bonds, the cell uses enzymes, and it has broken the process into discrete small reactions with a lot smaller activation energies that can be overcome by those enzymes. And therefore, at each independent step, the energy being released by the hydrolysis of the bonds between the sugar molecules can be harvested by the cell in activated energy carriers. At the end, you have the same product, carbon dioxide and water. The difference is that whereas in the environment all the free energy released as heat, it's not stored by the cell, in the cell, the free energy released by glucose, it's stored by activation carriers, and some of it will be lost as heat. Now, when we take food products, in this case carbohydrates, think about going to eat pasta at an Italian restaurant, those carbohydrates are found as complex polysaccharides, such as starch. During digestion, those complex polysaccharides will be broken down into simple sugars, like glucose. Now, once that large macromolecule, that polysaccharide, is broken down into smaller monomers of sugars like glucose, they can be transported across the membrane of the cell. And once they're transported across the membrane of the cell, through the process of glycolysis, you can break them down into pyruvate. As you can see here, in the cytosol, the glycosidic reactions will happen and generate some ATP and some energy carriers, NADH. Now, pyruvate in the cytosol will be transported across the mitochondrial membrane and through a different set of reactions be converted into acetyl-CoA. Acetyl-CoA, as you are aware, will be then brought into the citric acid cycle, where every single bond between the acetyl-CoA will be broken to generate CO2 molecules and highly energetic electron carriers. Those electron carriers are going to be brought into then the respiration electron transport chain. And when they donate their electrons to the electron transport chain, that will result in a proton gradient that eventually will be used for the generation of ATP. Oxygen is the ultimate electron acceptor, and that will be reduced to water. And water and carbon dioxide will be the byproducts of the reaction. This is happening inside the matrix of the mitochondria, whereas the electron transport chain will be happening in the membrane of in the inner membrane of the mitochondria. With that, I'll finish the lecture today. If you have any questions, please let me know.